Welcome students to chapter 19, which is going to focus on the second part of the cardiovascular system, the blood vessels. This lecture in particular is going to focus on part 1, which is looking at kind of vascular anatomy. And we're going to start by just looking at what the walls of the blood vessels look like, both in the arterial system and in the venous system. And then our little capillaries here. So when we talk about blood vessels, you have to remember that they are hollow organs through which blood will flow through the lumen. You'll see veins will typically have a larger lumen than arteries, but arteries typically have a much thicker wall than the veins. And then capillaries, of course, have the thinnest of the walls, as we'll see here in just a second. There are kind of 3 layers when it comes to the walls of the blood vessels. The inner layer that actually lines the lumen is known as the tunica intima. It has very typically tightly held together junctions in some sort of endothelial tissue, so nice simple squamous epithelial tissue, makes a nice smooth, kind of somewhat frictionless surface through which the blood will flow. That endothelium will have a little bit of connective tissue underneath, and then we'll often have a little elasticity in the artery, but not necessarily in the vein. The middle layer is known as the tunica media. It's mostly smooth muscle, but then also a little elastic tissue, particularly in the arterial system. So arteries tend to be a little springier than veins. And then the outermost layer of the blood vessels are the tunica externa, which is mostly fibrous connective tissue, which will then, in some of the larger arteries and veins, actually have their own blood vessels, what we call the vasa vasorum, which is literally blood vessels of the blood vessels. So the thickness of the walls and the amount of elastic tissue, smooth muscle, varies between arteries and veins, as we'll see. The thinnest of the wall of the blood vessel walls, of course, are capillaries, which is really actually just a little very, very thin tunica intima and typically lacks a tunica media and tunica externa altogether. This is the arterial system and the venous system. So here's your circulatory system connecting to the kind of other part of the cardiovascular system, your heart. So arteries are always leading away from the heart. Veins are always returning to the heart. In the systemic circuit, arteries are carrying oxygenated blood and veins are carrying deoxygenated blood. In the pulmonary circuit, of course, it's flipped, where the pulmonary arteries are carrying deoxygenated blood and the pulmonary veins carrying oxygenated blood. But in the systemic circulation, the arteries are leading away and carrying oxygenated blood, and the veins are returning, carrying deoxygenated blood. You'll also notice there's some lymphatic systems here. We're going to talk a little bit about capillary exchange a little bit later. And when that occurs, essentially fluid leaks out, but doesn't always make it all back into the circulatory system. And so we have the lymphatics here that can help pick up some of that. Leading away from the heart are what we call our conducting arteries or our elastic arteries. They have a lot of elastic tissue in them that keeps the pressure in this high pressure arterial system going. Those lead into the muscular arteries, which are actually the distributing arteries, the ones that actually go out to the stomach and the femur and the muscles and the organs, actually taking it out to all of those organs. The muscular or distributing arteries will then branch into these smaller arterioles. Arterioles are known as resistance vessels because they can change their diameter quite easily, which changes the resistance to flow, as we'll talk about in just a little bit. Leading into the capillary are the arterioles. The capillaries are where all of the exchange takes place, whether that's waste products, oxygen, nutrients, carbon dioxide. It's all moving into and out of organs in the circulatory system here at the capillaries. Coming out of the capillaries are small venules. Those small venules will then merge with smaller veins. The smaller veins will then merge with larger veins, which will eventually merge, of course, with the vena cava to take it back to the heart. Veins are known as capacitance vessels, in large part because most of the volume of the blood, the roughly 5 liters of blood in your body, are actually housed or are traveling through the venous system as opposed to the arterial system. So this is our typical setup: heart, arteries, arterioles, capillary, veins, vena cava, back to the heart. We've seen we've seen one already where the system is a little different. It's called a portal system, and we'll talk more about that when we talk about the hepatic portal system as well. But this is your typical setup in terms of how blood flows through your circulatory system. If we look at the walls of our arteries and see kind of what's in them, you'll see that these elastic arteries, they have the highest proportion of the elastic tissue. They also have elastic fibers in all 3 of their tunics, not just like in one particular level of the kind of the wall. They tend to have fairly large lumens, which means they're pretty low resistance. Blood moves through them pretty easily. They don't usually play a huge role in vasoconstriction and vasodilation. It's really mostly about the elastic tissue and then the elastic tissue's ability to kind of stretch as the blood moves through it, and then kind of recoil as the blood passes. So it acts like a pressure reservoir. Kind of being able to stretch and then recoil kind of keeps that high pressure going in the aorta and all of the branches off of the aorta. If we look at the muscular arteries, you'll see that, of course, the largest kind of distinguishing feature there, of course, is the smooth muscle, hence the muscular arteries. These are all the arteries that are actually going out to all of your body organs. Because they do have a ton of smooth muscle, they are quite active in vasoconstriction and vasodilation and kind of how that changes blood pressure, as we'll talk about a little bit later. The arterioles are the smallest in diameter. It's much tinier than what we're looking at in the elastic or muscular arteries. And these are our resistance vessels. When they change their diameter, whether they're vasoconstricting or vasodilating, that changes the pressure dynamics in the arterial system, which changes your blood pressure. The arterioles are what lead into the capillary beds, so they also control the flow of blood into the capillary beds. And so if you have, say, a capillary around your brain, if you want more blood to get to your brain, then the arterioles leading into all your brain capillaries have to vasodilate and get bigger, brings more blood into that area. And if they vasoconstrict and get smaller, then it's going to kind of shift the flow of the blood to that organ. The capillaries themselves are microscopic. In fact, some of them are so small that only a single red blood cell can kind of pass through it at a time. So we're talking incredibly tiny blood vessels. They have no tunica media, no tunica externa. All they have is tunica intima, and it's very thin, really just that one endothelial cell. We find capillaries in every tissue of the body, just about, with a couple of exceptions. Our cartilaginous tissues are avascular, our epithelial tissues are avascular, so those 2 tissues need other connective tissues near them in order to get oxygen and nutrients. Same thing with the cornea and the lens in your eye. They are also avascular. But pretty much every other tissue and organ in your body has capillaries in it, and so practically every cell in your body has direct access to a blood supply, and all the oxygen, nutrients, wastes, and stuff that come with it. The capillaries are where the exchange takes place. So again, whether that's exchanging oxygen for carbon dioxide, whether it's exchanging nutrients for waste products, whether it's hormones getting in to interact with the receptors, that all takes place through the capillaries. There are 3 types of capillaries, and it really is a function of how permeable or not permeable they may be. The most permeable are the sinusoid capillaries. The least permeable, of course, are the continuous capillaries, and they kind of tell you that in their name, right? Continuous. The continuous capillaries are the most common of the capillaries that we find. We find them in our skin, the muscles, lungs, nervous system, all over the place. They will often have very tight junctions between the cells, and so whilst things will move in and out of it through these exchange processes that we'll talk about a little bit later, it's not as easy with the continuous capillaries as it is with some of the others. And so something like the blood-brain barrier, where we want the blood to move and the blood to get out, but we won't want like toxins and things to get out of the blood vessels and into our brain tissue. That's much more likely to have a continuous capillary. Fenestrated capillaries are named for these little fenestrations or pores that are in their walls. They might have a little cleft in them, and so these clefts allow larger things to kind of get in and out of the blood vessel. Same thing with the fenestrations, the pores. We tend to find these where we want stuff to move more easily through in and out of the capillary, but we don't want we don't want to be able to drive a Mack truck through it. So if we're maybe talking about absorption of nutrients in our digestive tract, that might be a place where we would find a fenestrated capillary. If we look at the sinusoidal capillaries, just by kind of just looking at them, you can kind of guess that they would be the most permeable. They have very large intercellular clefts. The fenestrations are now not just little tiny holes, but big giant Swiss cheese. Really, really large pores and fenestrations. These pores and fenestrations and clefts are now so large that even big, large macromolecules can kind of slip through them. They tend to have these really kind of funky, irregularly shaped lumens, which kind of slows the blood flow through them. And so we tend to find sinusoidal capillaries where we want things to get in and out really easily, like for instance in red bone marrow, right, where we make all of our blood cells. We want those newly formed blood cells to get out of the red bone marrow pretty quickly and get into circulation. And so the bone marrow is going to have more sinusoidal capillary as opposed to something like a continuous or fenestrated capillary. This is what a capillary bed looks like. Again, our typical structure is an arteriole leading into the capillary and a venule leading out of the capillary. You'll see that on the arterial side, there's usually what we call a thoroughfare channel, kind of directly leading from the arteriole directly into the venule. And on either side of that thoroughfare channel are little smooth muscle sphincters. And if those smooth muscle sphincters, those precapillary sphincters, relax, then blood will flow through all of the capillary bed. So we want to really kind of soak that organ in the blood. Whereas, say we want to kind of say we're in a stress situation, our body doesn't really care about digesting food, so your digestive system isn't going to need as much blood as, say, your muscles, so you can run away, then these precapillary sphincters will basically shut, and that shunts the blood directly through that thoroughfare channel, and so it kind of bypasses the spreading out throughout the organ. So it helps us kind of route the blood to the organs where we need it. Leading out of the arteriole and our capillary, there's our nice little capillary, are the venules. They do tend to be quite porous. They might have a little smooth muscle, some of the larger venules, but generally just mostly a little endothelium, no elastic tissue in the venules. A little bit of elastic tissue in the veins. So when the venules kind of merge together, they make small veins then merge into bigger veins. Veins tend to have much larger lumens than the arteries, and so their shapes tend to be not as round. If you were to look at it in cross-section, they tend to have a little funkier shape in cross-section. A little bit of elastic tissue, but not as much as in the arterial system. So very low pressure system, but because the lumen is so much larger, they are our capacitance vessels, and about 65% of your blood is in the venous system as opposed to in the arterial system, or moving through the capillaries, the heart, or the pulmonary circuit. So you can see the largest amount of blood at any given time is in your venous system. So they're our kind of reservoir or reserve of blood, so our capacitance vessels. Because veins are kind of large diameter, low pressure, they have very, very little resistance in them, and so they have a couple of adaptations that the arterial system doesn't have. One of these is what we call the venous valves. Just like the valves in the heart, the venous valves prevent backflow of blood. They ensure that blood is moving in one direction. They are present in pretty much all of your veins, but are most abundant and most prevalent in the veins that we find in our limbs, so that the blood can get back up to the heart. We also tend to sometimes find sinuses, just like the coronary sinus that you guys saw with the heart, the dural sinuses of the brain. These are very flat veins that have extremely, extremely thin walls, much thinner than like a typical vein, that help kind of collect blood from an organ like the heart or the brain in order to then kind of shunt that blood back to the heart. There are a couple of additional factors that help the blood get back to the heart. It's what we call venous return. The first of these is the muscular pump. So here is our vein. You can see here's the valves in the vein, and the blood flowing up through the vein. So say we're in maybe the lower limb or the upper limb. Well, in and around all of our veins are all of our skeletal muscles, and when our skeletal muscles contract, that essentially acts like a milking action, kind of opening and closing as the muscles contract. That will close some veins kind of more distally, open valves more proximally, and that kind of milks the blood back to the heart. We also have what we call the respiratory pump. As you breathe in and out, that changes the pressure in your thoracic cavity, which tends to squeeze the veins in the abdomen and kind of vacuums almost the blood towards the heart. That's what I like to think of it as anyway, as a vacuum, kind of pulling the blood up the body from the abdomen. And then we also have venoconstriction. The veins themselves, under sympathetic action, can vasoconstrict, venoconstrict, since we're talking about a vein, and that can also help return the blood back to the heart. In terms of anastomoses, which are basically just connections between blood vessels, you probably saw a lot when you looked at the coronary system, because we tend to have a lot of anastomoses, these junctions in really important organs like the heart, the brain, some of the abdominal organs. And it basically is just allowing us kind of alternate pathways. So we can have arterial anastomoses in order to kind of make sure that the blood is getting out to different organs. Again, you're going to find those particularly in our really kind of vital organs. Those vascular shunts, those little thoroughfare capillaries and the channels that we see in the capillaries that directly connect kind of an arterial and a venule. That's a particular type of anastomosis. It's arteriovenous because it's connecting an arterial and a venule. And then actually in the venous system, there are lots of vascular anastomoses. It is very unlikely that if you have a blood flow problem, it's due to a blockage in a vein. Usually the more kind of damaging is if you have an occluded artery. And that's just a little bit of vascular anatomy. This will allow us to jump right into blood flow when we get to class, and vascular physiology, which is decidedly more complicated.